Okay, so this is your sixth week session, and in today's session, we'll be talking about the week six topics, which were talent technology, which is the transcription activator like effector nucleases. And in the previous session, we looked into the zinc finger nucleases. So these are the second type of nucleases. And in this session, we'll look into the basics of talent, how to design these talent for genome editing, and what are the application of these talents. And then we'll practice MCQs, and along with the content, we'll also look into the current state of literature, and then I'll also take your doubts. So let's talk about the thing which we can do with these talents. So the most important thing is the genome editing. So TALEN stands for transcription activator like effector nucleases. So as the name suggests nucleases, that means they can cleave nucleotides and they are powerful tool for genome editing. Because in zinc finger nucleases also we have seen that these nucleases they make double strand break in the DNA and then our DNA tries to repair it, and in the process of repair it can either lead to the incorporation of the gene or deletion of the gene. So depending on what we desire we can uh, get that result from these talons or these nucleases. It was developed in early 2010s as an alternative to zinc finger nucleases. So there were certain limitations of zinc finger nucleases due to which these talons were raised. So they have some advantages over the previous zinc finger nucleases. The precursor technology to CRISPR. So after talon, now we use CRISPR, but still important in certain contexts. For example, mitochondria. So for example, if we want to make any editing at the level of mitochondria, then talons are better as compared to the CRISPR-Cas9 technology. So in this session, we'll cover topics related to talon structure, how it works, its mechanism, how to design the talons, its applications, and its advanced uses. So this shows you the tail and talon tools. So as we know that these nucleases, these are not just a single unit. They are composed of different components. So one component is tail, which is the transcription activator like effector, and then it is joined to the nuclease. So nuclease is like a functional domain which is attached to tail, and then it acts like a nuclease, entire nuclease. So which is talon. And what we can do with this is like we can perform the work of transposases. We can, if we instead of adding nuclease, if we add transposase, then we can perform transposition of the genetic elements. We can visualize different things inside the cell, means how the genome is arranged, what are the genes where it is present. So by using talons we can specifically cut that region, and then we can extract it and we can visualize it, that which gene is present and what are the sequences which are present. It can also act as activators. So by adding different components to the tail unit, we can determine its role. It can also act as a repressor depending on which domain is binding to tail. Then DNA demethylase or methylase. Histone modifiers. We can also modify histones because we know that the histones are the proteins around which our DNA or so around which the DNA is wrapped. So the region where this chromosome or the DNA which is tightly wrapped, and when the histones are tightly wrapped, so that region is not allowed to express freely. That is the heterochromatin region. So by using the histone modifiers, we can modify it. We can either methylate it, acetylate it, or perform deacetylation. So in that way, we can govern that whether our gene should now freely express, can it get converted to euchromatin so that it can freely express, or do we want to turn off the gene? So in that case, we can modify the histone in such a way that gene is now tightly packed. It is in the heterochromatin state. So in that case, gene expression is repressed. Similarly, nucleases, which is the most important activity, which we will be going to discuss today. And then base editor. So we can also edit few bases which are present in our DNA. And then it can also act as recombination if we add the Cre-Lox, that system, along with the talon, along with the tail. So in that way, we can also bring about the recombination at the level of DNA. So these are different advantages. Now we'll talk about tail, so which is the basic component of talon. So we add nucleases to the tail unit. So tails are the proteins which are secreted by Xanthomonas plant pathogenic bacteria. And this Xanthomonas, so it is infectious in the plants and it is a bacteria. So it is delivered into the host plant cells by the type 3 secretion system. So keep these points in your mind because these are very important, that how this particular bacteria get into the host cell or into the plant cell. So there is a type 3 secretion system with the help of which it enters into the plant cell and inside the nucleus. So first of all, it will go through this cell wall and then it will enter into the cytoplasm. So at cytoplasm, it's ineffective because it works on the DNA. It makes double strand break at the level of DNA. So in cytoplasm, there is no DNA. So it has to enter into the nucleus. So it enters into nucleus and it binds to specific sequences which activate the host gene transcription. So it is binding here to this particular promoter region, which is complementary to that of the tail sequences. So in the later slides, we'll see that what is actually tail, how it is composed of, what are the sequences. So due to that specific sequences present on the this particular DNA or genetic element of the plant cell, it will do the specific binding to that element and it will lead to the transcription of certain genes, which are the host gene transcription. So related to plant cell only, the genes will get expressed. So the purpose is to reprogram the host metabolism to favor bacterial infection. So now the plant will synthesize those proteins which are important for the bacterial activity, for it to get control of the plant cell. So for infection, it will reprogram the host metabolism in such a way. So some tails instead trigger plant resistance genes, which will also activate the immunity of this particular plant. So some resistance genes can also get expressed. So for example, in this case, this is how the type 3 uh, secretion system. So with the help of type 3 secretion system, the xanthomonas will enter into the plant cell and it will deliver the tail. So it is not entering as a whole, but it is delivering its tail, which is the transcriptionally active like transcription activator like effector. So it will enter that particular unit into the plant cell. Now this is how it appears. Now what it will do, it will enter into the nucleus. It will specifically bind to the sequence present on the host gene or host genome, and then it will upregulate certain genes expression. So certain proteins or certain transcripts will get synthesized in the nucleus and then they will get exported to the cytoplasm. In cytoplasm, protein synthesis will take place. And ultimately, the proteins are specifically the metabolites which will help the tail effectors to further perform their function and infect the cell. So in this way, 25 other effectors can be synthesized and different resistance genes can be synthesized such as BS3. So this shows that what kind of genes can get activated. Now we'll look into the structure of tail, that how it actually looks at the level of molecular level. So its central feature is that it has a central repeat domain, which is composed of 18 34 amino acid repeats. So these red boxes which you can see, so these are the repeat sequences and there are total 18 such repeats and each repeat is 34 nucleotide or 34 amino acid in length. So in this we are talking about the protein structure, protein sequence. So this is in the terms of amino acids. So there are 34 amino acids and these 34 repeats will come 18 times. So this is the main feature of this tail, that whenever you find such larger repeats occurring 18 times and they are present consecutive to each other, that means next to each other. So this particular structure belongs to the tail, that is TALE, and each repeat forms a 2-helix bundle with a loop at position 12 and 13. So this is how the structure is formed, that there are two helices, 2-helix bundles, and then there will be a loop at position 12 and 13. So these loops, they represents this particular structure of each repeat, and then repeat variable residues. So there are some repeat variable residues which are also present. So the two amino acids in the loop that determines which DNA base is recognized, because these talons or these tails, they will bind to the genomic sequence of the plant cell or the host cell. So in that case, the sequence which governs to which nucleotide sequence it has to bind, so that is governed by these two amino acids which are present in the loop region. So specifically the 12th and 13th residue, which is making the loop-like structure, so that structure will govern that to which nucleotide sequence it will bind, and that particular sequence which is variable, it is referred to as repeat variable diresidue because there are two residues. So this N and G at position 12 and 13, they are referred to as RVD or repeat variable diresidues, and they will decide that this particular tail element to which genetic component it will bind to. So for example, if it is NG, then it will bind to T, which is the thymine residue of the plant genome or any host genome on which you want to target it. So it will bind to thymine. If it is Ni, so it will bind to adenine. If it is Nn, then it will bind to Ga. If it is HD, it will bind to cytosine, and if it is NS, then it can bind to either A, G, C, or T. Now, why it is important to keep these things in mind? Because in your tail sequence, you have 18 repeats. So that means in all these 18 repeats, you will have these two residues present across all. So in this way, in the end, you will get 36 such amino acids. And these 36 amino acids, so each, means a pair of them will bind to a single nucleotide sequence. So that means these 18 repeats having diresidue for specific for each one, so they can bind to 18 nucleotides. Now, if we want to design them in such a way that I want to target a specific region for which I know the sequence, so I can design TELN according to that. For example, I want to bind a sequence which is TGC. So in that way, in one of the repeat I'll keep NG, in the other I'll keep NN, and in the other I can keep HD. So this is important if you want to design a specific talent, so specific tail specifically. So we can design this repeat region. We can assemble it and we can design specific tail for specific region in the genome. So for that reason, it is important to know that what is the specificity of this RVD dimeric sequence. So its C-terminal region contains nuclear localization signal and acidic activation domain. So this is the NLS, which is the nuclear localization signal, which is important because as soon as the tail will enter into the cytoplasm of plant cell, now it has to enter into the nucleus because it want to bind to the genome of the organism. So for that particular region, it has this NLS sequence or signal, which will help it to transport into the nucleus. And then it also has the acid activation domain, which is this one. So this region, it is important to recruit the host transcription machinery. So it will activate the transcription of certain genes, which are essential for the bacterial infection to proceed. Now we'll look into the RVD code. So as we discussed that each of these repeats where there is 12th and 13th positions, that is the loop which is joining two different helix bundles. So this loop has two amino acids which determines that to which nucleotide sequence it can bind. So one repeat, one base. This is the rule because each repeat has two these amino acids, 12 and 13. So they 12 and 13 will bind to only single nucleotide. That means total we have 18 repeats. We can have more than that also. It depends how we are synthesized. So that will be our artificial tail. So each repeat can bind to one base. This is the rule. And common RVDs are Ni, HD, Ng, Nn, or Nk. And these are the specific nucleotides to which they can bind. And the other RVDs, which we artificially design, so they can vary, which we have seen in the previous example that Ns can also bind to all of the N, A, G, C, T, but in that case, it is not specific binding. Specific binding means only it can only bind to a single nucleotide. So in that case, we know very well that it will only bind to C. But in case of NS, it can bind non-specifically to either A, either G, either C, either T. So it depends that what uh, di-amino acid we want to keep. So this um, simple modularity make these tails easy to reprogram to bind the new DNA sequences. So it depends that to which DNA sequence we want the tail to bind. So in that way, we can design the tails. Now this is also the example that, as you can see, there are multiple such repeats. So all these repeats which are present inside the tail sequence, and this is showing that how the structurally it looks like. So there are two helix bundles. One is the short helix, which is from 1 to 11, and then there is 12-13 loop, means 12-13 region is falling in the loop. And then from 15 to 33, there is another larger helix, and then there is another turn. These residues are contributing to turn, the 34th residue. So this is the structure of the tail, that is transcriptionally transcription activator like effector. Now we will see the function of tail in plants. So example is that rice infection by the Xanthomonas oryzae, because we have seen that tail is generally obtained from the bacteria named Xanthomonas, which is the pathogenic in nature. So these tails, it binds to the promoter of sweet genes because we know that this particular tail, it will bind to specific promoters or specific sequence. So generally, it binds to the sweet genes promoters. So as it binds to the promoter, it has an acidic domain, which we discussed. So that is responsible for the transcription of the host gene. So this will lead to the transcription of sweet genes, which induce sugar flux from the cells. So this sugar will be used by the Xanthomonas oryzae. It will use it for the infection of that particular host cell. So the sweet gene will get expressed. They will synthesize more and more sugar, and this sugar will be exploited by the bacteria. Then there could be some host resistance genes which can also activate. So some plants carry the executor R genes that are activated by the tails, which will trigger hypersensitive response, which can lead to the cell death to stop the infection. Because if Xanthomonas is exploiting the plant machinery, then plant has also defense mechanisms against that particular Xanthomonas. So in other cases, it can also synthesize some of these resistance genes, which can kill the pathogen, which can kill these Xanthomonas. So in that way, it can stop the infection. So tails therefore function both as virulence vectors and as the immune triggers. So it is not just leading to infection, but it can also activate the defense mechanism of the plant cell. So in that way, it is triggering the immune response depending on the host genetics. So it depends on the genome of the host. So not in means in not all the plants you will see the expression of executor R genes or the resistance genes. So it depends from genome to genome. But in this case, rice infection, generally the tail binds to the uh, these sweet genes. So this is what was observed. So all these experiments, these are the experimental results. Means experimentally these things were observed, and later on scientists thought that okay, we can also explore different roles by using the tails, because as bacteria is using it for the infection, so we can also use these tail elements, and then we can repurpose them for our function, means on our desired function, so we can get few advantages from them. So this is how the discovery started, and this is how artificially scientists thought that we can design our own artificial tail elements and then join it to nuclease and perform certain functions such as gene editing or all other things. Okay, so this is also showing the same, that we can also construct the tail in such a way that instead of activating sweet genes, it can activate those genes which could increase the yield of the plant. But this thing will be performed artificially because we are not using Xanthomonas to infect it. We are doing it at means experimental level. So to just increase the yield. So in that way, we can design them in such a way that it specifically trigger those genes which are essential for the increase of the yield. So this will be the artificial role. Now, how this binds to the DNA? So there is other mechanism. So the CRD of the tails wrap around the major groove of the DNA in the right-handed superhelix. So this is the structure of DNA, which is shown in the reddish orange color, and then it is wrapped around. So generally, any of the nucleases or elements which interacts with the DNA, it will always interact with the major groove of DNA. So keep that thing also in your mind. So each RBD in the repeat will interact with a single nucleotide, which we have already discussed, residue 12 and 13. So residue 13, it makes the direct hydrogen bond with the DNA base. Now, what are the means the chemical events which are taking place? So in that case, residue 13 is making the hydrogen bond with the base of DNA. So DNA base here means the nitrogenous base, such as adenine, guanine. So with them, it will make hydrogen bond, and residue 12 is important to stabilize that structure. So as you can see how it is looped around the major groove of the DNA. So this loop-like structure, this is favored by, this is stabilized by residue 12, and 13 is responsible for the binding. This modular binding form a protein code for DNA recognition. So protein code here we mean that for each repeat, we can have the specific code for the residue 12 and 13. So this is what they are calling as protein code. So in that way, to bind to specific nucleotide sequence, we can design this residue 12 and 13 for each repeat in such a way that it will behave like a code, protein code. So for each of the DNA sequence, this code will also vary. So this shows you that if these are the amino acid sequences present in my tail, then to which DNA sequence it can bind. Okay, and this is the further diagram which is showing that one of the amino acid at the RVD, which is at position 13, it is making the hydrogen bond with this particular base of the DNA. So this is a small part of the picture. This is not the complete picture. So this is just a small part in which this is the DNA helix, which is shown here. And this orange colored chemical structure, it belongs to the purine. So this is one of the purine, because we know that purines have the double chains, double rings. So this could either be adenine or guanine, and one of the residue of the RVD, one of the amino acid, which is interacting with this particular nitrogenous base, whereas the other residue is responsible to stabilize this loop-like structure so that it can stably bind or stably make the loop-like structure around the DNA. So that is the main purpose of these RVDs, which are the variable residues. So at position 12 and 13 in each repeat, and there were total 18 such repeats present in the tail sequence. Now the next thing is search versus recognition states. So tail binding is dynamic. There are two states which exist for the binding of tail to the genetic element in the host. So first is the search state, or which is also known as the search mode. In this case, the superhelix is loose and extended. So in that case, the pitch is around 60 angstrom. So pitch simply means that what is the length or what is the distance of one single turn. So if we measure that, that is 60 angstroms, and it allows the 1D sliding and 3D hopping along the DNA to find the target. So now, when our tail is that particular effector molecule, when it enters into the nucleus, then it will not directly go and bind to the site. It firstly it will search for that site. So it will non-specifically bind to some region and then it will slide. And it can also do 3D hopping. That means it can simply glide, or like, means in hopping we means like smaller jumps it can perform. So in that way it will search the entire DNA to find the target site where this RVD domain, RVD sequence, which is present in between the central repeat domain, they will decide to which nucleotide sequence to bind. So that state where it is searching for that particular target site, that is known as search state. Then comes recognition state or the bind mode. Now, as soon as it reaches that site, then it will compact the superhelix. So this pitch will be 35 angstrom. So when we were looking at the secondary structure of the proteins, then we were discussing about the pitch. So similarly, this is the protein sequence. So it is also having a bigger structure. So in that way, when it was searching, then it was loose superhelix, which was around 60 angstrom. But as soon as it finds the target site, so now it will begin to compact. So as you can see, this is quite loose. There is a longer gap. If for a single turn, if you measure the length of a single turn, it is long. It means this is relaxed and this is extended alpha helix. Whereas as soon as it finds the target site, it will begin to shrink. The pitch will decrease. That means that one single turn will have a smaller distance. So this length decreases. That means it becomes compact. So this compact superhelix state will achieved as soon as it bind to the target and will form a stable base specific interactions, because we know that each of the RVD sequence will bind to a specific nucleotide position. So it will make a base specific interaction. So this switching ensures the rapid target search with high specificity, because if it is compact from the beginning, then it will take a long time to find the target site. So that's why in the search mode, it is extended superhelical structure, and as soon as it finds the target site, it will get compressed. So this particular switch in its structure is helping it to perform the faster searching and also high specificity due to the RVD amino acids. So now what are talents? So these are the engineered proteins, because till now we were only looking at the tail. In bacteria, Xanthomonas, so it only had a tail, which is the transcription activator like effector. And its role was to activate some of the genes of the plant cells to exploit or to get nutrition for its infection. But what we have done is we have designed talons, which means tail along with the nucleus. We have added the nucleus component to the tail. So these are the engineered proteins. That means we have artificially synthesized them or artificially engineered them. It has a DNA binding tail domain which is fused to the fork1 nucleus domain. So this in zinc finger nucleases also we have seen that there are zinc finger domains which are specific to bind to the DNA. So similarly in this case also it has DNA binding tail domain, and then we have fused fork1 nucleus domain to it. So when these two talons it binds on the opposite sides of a DNA strand, then fork1 will dimerize. So this fork one, it if it do not dimerize, then it cannot bring about the cut. It is unstable, so it needs its dimeric form. It needs another unit of fork one nuclease to bind and then bring about the double strand break. So in this case also the same machinery works here, that the fork one will dimerize and it will bring about the double strand break in the DNA helix. And then after the double strand break has happened, so our cell will begin the repair of the DNA. So in that case, there are two pathways which the double strand break can achieve. So one is the error prone pathway, in which it will use the non-homologous end joining method, in which the deletions could happen in the gene wherever the cut has been produced. Some genetic elements can get lost, or some small indels can be performed. So this can also lead to gene knockouts. So gene knockout means if we simply remove that gene. So it depends that how we are performing, how we are guiding NHEJ, so that the gene knockout is brought about in the that particular DNA element. And then another pathway is this homology dependent repair, in which we need the another complementary strand to that particular DNA strand, so that it can synthesize the another complementary region where the loss has happened, where the break has happened. So in that case, we can either repair the faulty gene after the double strand break, or we can also insert the new gene, or we can also bring about the single nucleotide polymorphism SNP by adding the new nucleotide. So in that way, we can bring mutation, which can help us, or it depends that what we want. So that mutation could also be disease specific. So if in case we want to study the disease model, so in that case, we can keep SNP such that it leads to the disease state. So in this way, by the help of homology directed repair, we can either insert some element, or we can either invert it or correct it. Whereas in NHEJ, it leads to the indels or knockout of the gene. So these mechanisms we have already discussed a lot from previous sessions also. Now there are some alternative architectures. So we know that fork1 is the standard nuclease which is generally used for zinc finger nucleases and for the talons also. But there are some other nuclease fusions also which were explored by the scientists. So restriction enzymes such as PVU2. Then there are some meganucleases which are ITEVI or IANII. Then there are some nicases which produce the nicks in the double-stranded DNA. So they are like mut-H, truncated ITEVI, which introduces single-strand breaks and it lowers the off-target risk. But after performing all the experiments with these types of endonucleases or the restriction enzymes, scientists have observed that the fork one is the one which brings about better functionality, which is most commonly used. So it's robust also, and but it requires the dimers. That means it cannot perform single-handedly. We also need the another fork one unit. So in this way, there are two units of fork one which will dimerize, and then they will break the double-stranded DNA by bringing about the double-strand breaks. So after comparing all of these nucleases, fork one is the one which is most dominantly used. Okay, now we'll look into the assembly methods, because we have seen that we have engineered these talents. Now we also want it to specifically bind to specific region, because if I want to perform gene knockout, then I don't want to perform a random gene knockout. I must have some gene in my mind which I want to knock out. And why these knockout experiments are important? Because if we want to see that what is the function of the gene, then to see that function, we need to knock it out, so that if the phenotype, there is change in the phenotype, then we can judge that okay, if this gene is removed, then this is loss of this phenotype is observed. So the gene is important for this function. So that for that region we perform the knockouts. So now I know which gene I want to knock out. So similarly, I can design the talon which can specifically bind to the promoter of that gene or some region of that gene, so that I can make a double strand break in that region and I can knock out that specific gene. So to synthesize the talons, there are assembly methods, because we know that these tails or the transcription activator like effector molecule, it has multiple repeats, and each repeat has the RVD sequence which is specific to the nucleic acid sequence it will bind. So in that way I can change those repeat sequences in such a way and join them, so that I can decide to which nucleotide sequence it should bind. So for that region, to synthesize those specific tail molecules, we use assembly methods, in which we assemble the repeats in such a way that they can target the specific sequence of the genome. So the challenge of this is that assembling many near identical repeats is a challenge. And then the solution is that we can design the specialized cloning systems where we can clone it and we can check that which of them is more specific. Okay, so the methods which assembly methods include Golden Gate, which is the modular cloning system, then Flash, which is the solid phase ligation system, ICA, which is the iterative capped assembly, DIY kits, which is simple in-lab protocols, and STAR, which is the Gibson-based rapid assembly. And we will one by one go through all these methods. But the main thing which you should keep in mind is that in all of them, if you could see, then uh, this HD, this shows you the 12th and 13th residue of each repeat. And these different boxes, these are the multiple repeats. So P1 is the first repeat, P2 is the second repeat, P3 is third repeat, P4 HD is the fourth repeat. So in this way, we have different such repeats. And now they are saying that the problem is that if the sequences are nearly identical, then how to decide that what should be the composition of these repeats. So in that way, we can design multiple such tail molecules, and then we can use the plasmid to clone it to make multiple copies of it, and then we can specifically check that which one of them is better. And then we can perform the transfection mRNA synthesis for them, and we can add the fork one to it, because in the plasmid construct itself we will add fork one in such a way that we are joining the tail elements along with the fork one nuclease. So this is generally to check that which one of um, these sequences, when we are assembling these smaller repeats, so which repeat sequence is better and which one to decide, which one to choose. So for that reason, these experiments are done in the lab so that we can decide. Now we'll talk about the Golden Gate assembly. This is the first way in which we can assemble these small repeats. So it uses type 2s restriction enzymes. So we have seen three main classes of restriction enzymes. So there is type 2 type of restriction enzymes, and how these types are decided. So it depends at which site they cut. So if they cut at the recognition site, then they're generally type 2 restriction endonuclease. If they cut far away from the restriction site, then they could be type 1 or type 3. And then also it depends that is there any specific region how after how much sequences it will cut. So that will enter into the type 3 endonuclease. So similarly, in Golden Gate assembly, we uses these type 2S restriction enzymes such as BSAI or BPI1. So you can either call them as BSA1 and BPI1, or I depends on you. So these are the Roman uh, numerals. So that cut outside the recognition sites. So in this, it cuts the outside of the recognition sites, and it allows, because these are type 2S restriction enzyme due to which it is cutting outside the recognition sites, and it allows the seamless and directional assembly of the repeats. And it is a pre-made library of 68 modules for any 17 base pair target sequence. So in this case, there is already available 68 tail repeat modules, which are already available. Modules are like smaller chunks which you can join them together. So means these repeats, because we have seen that in the tail there are generally 8 repeats. So in this module there are total 68 such repeats which are already synthesized. Out of them, you can select the specific 18 such repeat sequences which are specific to 17 base pair target sequence. So they're already designed, and you can simply use them. You can simply join those modules together to decide the target sequence according to your interest. And it requires the inclusion of repeat 0, which is T preference, and half repeat 17.5. So in this case, the first repeat or repeat 0 is the T, so T preference. And advantage is that it is efficient, cheap, and accurate. Why it is cheap and why it is efficient? Because we are already having the pre-designed such tail repeat modules, so we can simply pick those modules and join them in such a way that it can bind to our specific target sequence. Because for each of those, as you see here, it's written A, C, G, A, T. So what it means is that it is showing you the RV sequence which is specific to adenine, and this is showing you the RV sequence which is specific to cytosine. So in that way, we can decide if my sequence is composed of, for example, C, T, A, G, then I will pick up this blue one first, then I will use the red one, C, T, and then for A, I'll pick green one, and for G, I'll pick this yellow one. So I'll join them in that particular order so that it can bind to my desired sequence. So that is what is shown here, that how you can assemble it in the end. And that's why it is quite efficient and it is cheap because you don't have to design it yourself. It is already present in the repository, so you can simply pick it from here. And it is also accurate because these 68 repeat modules, they have experimentally tested that how well they bind to the specific nucleotide sequence. So all of this is already known. So this type of assembly is known as golden gate assembly. Now next one is the flash and ICA. So flash stands for fast ligation based automatable solid phase high throughput. So this is a high throughput system. High throughput means if you want to perform it for the longer sequences, then that is known as high throughput, which is accurate and which can be automated for the larger system. So that is high throughput and it has a robotic system. It has fast ligation on the solid support. So this is the archive of 376 pre-assembled tar effector repeat units. So in this case also there is an archive, means already this is known, 376 pre-assembled units which can be bind to either 1 mers, 2 mers, 3 mers, 4 mers. So these mers specifies that how many nucleotide stretch it can bind. 1 mer means it can only bind to one specific nucleotide sequence. 2 mer means it can bind to two nucleotide sequence. 3-mer means in my host genome, if there are 3 nucleotides and I want my module to bind to 3 nucleotides, then I will select this 3-mer. Similarly, 4-mers. And then we can join them together. We can assemble them and then we can check that whether they are, means after joining them, we can use the host, that particular cloning systems in which we can add the fork nucleases to them and then we can test them experimentally that whether they are binding to those specific regions which we are thinking of, those specific nucleotide sequences or not. So that checking can happen in the end. But now in this flash case, these are already known, so we can simply join them together. And the one of the advantages that they are quite high throughput, and in this case, formers are also available. So means using four separate one mers, we can simply use one single former. So this is also known. So this can reduce the load on us. So that is the advantage here. And then there is ICA, which is the iterative capped assembly. So it add the repeats one by one on the solid support. And then capping oligos block the incomplete products. So the advantage of iterative capped assembly is that in this case there is a solid support. So now, okay, so in this case also when we are assembling them, then they are bound to a solid support, which is here in this case it is a biotinylated DNA fragment which will bind to my this uh, particular support, this solid. So this biotin will bind to this. This could either be a streptavidin or something to which my biotin can bind. So this is bind to a solid support and then we are joining the other modules or smaller such units and then we are making a bigger tail molecule. So similarly in this case in ICA, similarly we have the beads, we have a solid support and now we are binding each such module one by one. And what I'll do, I'll bind one such module and then there is a capping process which will take place. So what capping will do, it will hinder the other such modules to attach to it. So it will make sure that the modules are assembled one by one. So as you can see here it's A and AB. Similarly it's bound and then C will bind here and then it will increase. So each time the capping will ensure that the oligos which are the incomplete products, they are blocked. Okay, so one more thing which I want you to focus on is that, for example, when we are adding A, which is my first level of means when I'm assembling it, I will add multiple units. So first of all, when I'm adding my units, so in that case, no capping is required because there is nothing at the base. But when I'm adding the second unit, then what can happen is that, for example, in this particular case, as you can see, no A is bound. So now if my B will bind, then in the end, the tail which I will get, it has lost the A, which is the first unit. So in that case, it will not bind to the specific sequence. So I don't want such faulty assembled tail units. So to avoid that case, the oligo will bind here so that the faulty assembly does not happen. Similarly, in this case, now it is bound to avoid the incomplete products. But now AB, now if I'll add C, then there is two conditions. It can either bind to A or AB. So if it will bind to A, then it will give a faulty assembled tail unit. So I want it to bind to just AB and not to A. So to block this, we use the capping. So now what capping will do? It will block this site. It will not allow C to bind to the that particular unit where only A is present. So this is the purpose. This is why ICA is used, so that we can block the faulty assembled products or faulty assembled tail components. And these both are suitable for building the large libraries of talents quickly. Okay, so this is whole system, means whole method of IC and flash. Now next is the STAR method. So STAR stands for simple tail assembly reaction, and it is based on the Gibson assembly, which is the isothermal DNA assembly, and it builds entire tail arrays in around 8 hours. And it only requires a small set of pre-made fragments, and it is used for the labs without the specialized cloning tools. When specialized cloning tools are not present, then generally STAR methods are used, and this is the representation of STAR method. So here are all the steps which are shown, that we already have the 68 part libraries in which already pre-designed modules are present. So it will take 30 minutes in which former assembly, which is the first Gibson reaction. So when we were talking about this particular flash, then we discussed that there are 1mer, 2mer, 3mer, and 4mer also present, in which at the same time I can get 4 amino acid RVD sequences, RVD units, which can specifically bind to 4 nucleotides. So if I join 4mers together, then that will be fast instead of adding 1mer 4 times or 8 times. So that is what is seen here, that they use 4mer assembly, which is the first Gibson reaction, and after 30 minutes, the plasmid safe DNAs are used, which clean up the reaction. And after 60 minutes, PCR amplification is done, which will enrich these 4mers. And then after another 60 minutes, SPH1 digestion will take place, which will generate the blunt ends. And after 45 minutes, this SPR1 cleanup will take place, in which we'll select the size and we'll clean up the 4mers which are additional, which are not required. And then after 60 minutes, full tail assembly will be performed, which will be the second Gibson reaction. And in the end, bacterial transformation, in which tail plasmid will be expressed in the bacteria. Okay, so this is the whole how the process is, what is the mechanism of generation of the assembly of the tail. So you don't have to go into the detail how it is taking place, but these are still some steps. If you're interested, you can go into the details, and there is a paper for which I have given the link. So in that they have clearly explained the stepwise reactions. But from the overview, the thing which you need to keep in mind is that in all these steps, in all these methods, what you are doing is you are assembling the units in such a way that you can design the tail component which can specifically bind to the nucleotide sequence. So you are generally changing the repeats present in that particular tail unit. So you can altering those repeats according to your desired target sequence. Now there are some computational tools also which you can use. So before building tails, we can use these computational tools which will tell you that what tails will help you to bind to this particular sequence. So instead of doing it experimentally and then checking that whether my repeat unit is binding to nucleotide or not, you can design it computationally, and then after computational design, then you can go for experiments or assembly. So design softwares are tail NT, which is the tail effector targeter, then tail plasmid assembly tools. Then there are some prediction tools also, which are target finder, talgetter, talviz, and then off target analysis, for which the tools which you can use is prognos, sapta, telenoffer, mojo hand. So all these things ensures that you have high on target activity and low off target risk. So now why it is that there is off-target risk? Because we all know that genome is long, very long, and it also has different nucleotides which are present across entire DNA, and there are only 4 such nucleotides, means 4 purines, pyrimidines. If we count them, then we have only 4 unique nucleotides, and which are combining in such a way that it is designing the entire DNA sequence. So there are chances that when I'm designing my tail sequence, if there are some repeat regions or such regions in my DNA, then my tail will get confused that to which site it has to bind. So it can also bind to some off-target site. So that is the risk which is there. So in that way, we need to keep those things in mind to avoid that low off target risk. But still, in the end, also there is certain risk which still prevails. But still, by experiments, the scientists have figured it out that what of the sequences, what of the talent units, if joined together, then more of the on target activity is achieved and less of the off target activity. So for that particular purposes, we need experimental validation, whereas we can synthesize the talents by using the computational tools. So that is the first step which we can keep in mind, which with which we can fast up the process of the tail molecules. So this is the flowchart of Mojo hand, which we have seen here, which is the off-target analysis. So it takes gene IDs from the NCBI and then contig beginning and end indices from NCBI nucleotide database and the files taken from the FASTA. FASTA is generally giving you the nucleotide sequence, and this is telling you the beginning and end of the, for example, I want to target a specific gene and I have entire genome. So where to get the sequence of that gene, how to get to that region in the entire chromosome. So for that particular purposes, I can look into the beginning and the end indices, because chromosome is a bigger structure. Chromosome is like a big DNA molecule in which multiple genes are present. So to find from where my gene is starting, to get that serial number, its ending and beginning. So for that we use contig beginning and end indices from NCBI. We get the ID of that particular gene also from NCBI and its file, that what is the sequence of that particular gene. So we will put it into the TAL site finding algorithm. Now what it will do, it will find that particular gene sequence in the entire genome, and then restriction site identification. So I will also identify the restriction site so that I can cut there, I can insert something into it. So restriction enzyme site is identified, that which site is present, and then we will report is made in the form of text or either CSV, which is the comma separated values, and then we enter the RVD into the spreadsheet. So in that we also need to enter the RVDs, and it will customize the talent recipe, that how those RVDs should be arranged so that that particular talent will specifically target my gene of interest. So in that way, in the end, talent assembly reaction will take place and it will give you the sequence of talent. Yeah, so I have kept these tables from this particular paper. So if you're interested, you can scroll through this paper and you can read it. So this is the detailed version, uh, each specific detail is kept regarding all the bioinformatic tools which you can use to design the talents, that how you can design it and what are the tail effector tools. So for the details, you can go through this, and then some tail effector nucleases tools. So this will also add the nuclease function into it. It will also tell you that how the fork one, where the break will happen. So all that information will also be given by using these tools. Okay, now there are the web pages or web servers of each of those tools. So this is how the tail effector targeter looks like, which has different options for you. So you can choose any of the options according to you, what you want to see. Either you want to design the arrays for the tail effector nucleases to target your DNA sequence, because means whenever we are doing knockout studies, so not everyone is working on the same gene. So it can happen that you have some unique DNA sequence for which the talent is not available. So in that way, you can specifically design those talents by performing the assembly of the smaller already designed RVD domains. Then talent targeter. So means based on what you want to target, which is the old version, then effector targeter, which design a single tally effector to target your DNA sequence. This is the newer updated version of single tail targeter. Then target finder, pair target finder. So all are these functionalities of this particular tool, which is the tail NT. Similarly, there is tail plasmid assembly tool. So I've also given the site. So if you want to explore these tools, then you can go through these sites and you can see what you can do with these tools. Then this is the tal getter, that you can write any job name. For example, if I'm working on my protein kinase, then I can simply write protein kinase, and then I can input the sequence of that particular gene. I can also enter the FASTA file, which I've already downloaded from NCBI, and then I can also give the RVD sequence, and then upstream and downstream offset. That means in the genome from where the gene is starting and where it is ending. that is the offset, and then maximum number of target sites, and then computation of p-values. So all these informations need to be put into the targeter, and in the end it will analyze that how well it can do binding, and where the cut will be happening, how the gene knockout will take place. So all these things can be checked. It can either be analyzed, or we can also design the talents. So, okay, so then there is eTalent. This is another software which is used to design and evaluate the talents. So we can evaluate it, that where the cut will happen and what kind of changes I can do with it. Okay, now there are some journal application of these talents. So those applications we have discussed a lot many times, such as gene knockouts. So how to knock out the genes by performing the indels, either insertion or deletions. Then gene knock-ins by using the homology dependent repair, and then modifying the embryo, stem cells, and somatic cells. So these are general applications of these talents. So I will again go into the mechanism, how it happens. So we have a double helical structure of DNA. Now there are designer nucleases. Why we are calling it designer? Because we are engineering them artificially. They're not originally present. We are artificially designing them. So in ZFN, we have zinc finger domains which specifically bind to the DNA sequence. So we are adding nuclease so that we can specifically target that particular gene of interest and then with the help of nuclease, I can bring about the double strand break. Similarly, in talons also, the tail unit is joined to the nuclease. And then in the next session, we'll talk about CRISPR. So in CRISPR also, the thing is same, that there is some guide RNA which specifically bind to the target site, and then with the another enzyme, it will bring about the double strand break. So what happens after the double strand break takes place? Then our machinery need to repair it. So it can either go through NHEJ or HDR. Now, if I am designing it artificially, then how I will decide to where to proceed. So it depends on my interest, what I want to do, whether I want to do knock-in or knockout. If I want to do knockout, I don't have to do anything, and it will directly enter into NHEJ. But if I want to do some knock-in study, then I need to enter the plasmid DNA or some DNA which has the homology arms, so that homologous recombination can take place, so that I can add the region of interest into my DNA where the Brock has happened. So what I can do, this region will be my homology arm, and between the homology arm, means in both of them I need to have some sequence which is same in both of these uh, DNA, in both of these chromosomes, or you can say these strands. So if there is the same sequence present in both of them, then what it will do undergo recombination. So between those two homology arms, I will keep something in between which can further enter into my target DNA. That is how knock-in is done. That means you are inserting the new gene, and you can also repair the gene which was previously faulty. So after the double strand break, what we can do is that which gene we want to remove. So the homology arm can be decided by us. So what we will do, we will design this particular DNA in such a way that it can specifically remove that particular faulty gene, and in this plasmid DNA, we will keep the repaired gene in between the homology arms, so that the recombination takes place and the faulty gene is exchanged with the correct gene from the single strand or plasmid DNA. So that is how the correction of gene can be done. And then ultimately, we can clone them into the animal cells by injections into the embryo. These are the later techniques, that how after gene editing, after correcting the gene, we can enter it into the host cells. Okay, now other than these editings, other things which can be done is transcription factors. It can act as base editors, recombinases, and epigenome modifiers. So in the case of TELN, we were adding the nuclease unit to the tail. But other than nuclease unit, I can also add different units, and I can change the functionality of these tail elements. So these are some of the examples that when we add nuclease, it can lead to the insertion of gene, deletion of gene, or modification of gene. If we add activators, then that activators could either be VP16 or VP64. So instead of binding using fork1, we use VP16 and VP64. They can activate the genes. Then by using transposase, so we can perform certain mutations which can lead to the transposition of the genetic elements. Then if we add the demethylase unit, which is LSB in this case, so it can lead to gene suppression. So demethylation will lead to the suppression of genes. Then recombinase, so instead of nuclease, if we add recombinase, such as REC, then it will lead to either gene insertions or stacking, or inversions can also take place. Then if we use repressor, such as CRAB, SID, then it can repress the gene. If we add methylase, such as TET1, then it can change the gene expression. It could either lead the expression of certain genes and alter the epigenetics. So epigenetics means that when something gene is exposed to some environment, how it behaves. So similarly, it will change the epigenetic because I'm using the methylase unit, which is doing methylation. So I'm changing something in its environment. So it is also altering its epigenetics. Okay, then there are certain studies. So first, Talon knockout rat in 2011. So this study tells you that how the knockout rat has been developed by using these talons, by performing the microinjection into the embryos of the rat. And in this, the target was rat IgM locus, because we know that tails are binding to specific sites. So in this case, the target site were the IgM, which is the immunoglobulin M, which is like we have the antibodies. So the IgM is specific type of antibody, which is important for the B cells. So talon editing produced the rats without the mature B cells or antibody secretion. So in this case, what they have done is they have used the talon, and in talon FOC1 nucleases were present, and they specifically target this particular gene locus. And after the double strand break has been happened, so it led to the disruption of this gene. This without the IgM synthesis, it led to the immature B cells or antibody secretions. So there was no antibody secretion. So this tells you that how we can target certain genes, how we can bring about the disruption in the genes using the talons. This is the first demonstration which tells you the gene knockout mammals can be generated using these talons, and this opened the path for disease models. So what ultimately we got from here is that we know now that what is the function of this IgM. This is interfering, this is responsible for the mature B cells and antibody secretion. So if something happens in this particular gene, then it can lead to the uh, compromised antibody secretion, and it can lead to immature B cells. So this is how we can explore disease models. By bringing about certain mutations, we can study that what could be the impact of these mutations, because there are many diseases such as cancer and other such incurable disease, and we don't know what is the exact reason. So with the help of these knockout studies, by performing certain mutations, we can study, we can link that, we can link the phenotype to the genotype. So that is what the purpose of these knockout or knock-in studies. And then there is another study on the zebrafish, which is the zebrafish skid model of 2016, performed by Jung et al. And for all these studies, you can click on these links. I have given the links to these papers from where I've got the information. So they targeted the PRKDC genes, which are the DNA PKs, protein kinases sites specifically. They result in the immunodeficient zebrafish, which are lacking T and B cells. And this mimicked the skid disease, which is the severe combined immunodeficiency. And the application is xenograft model for cancer research. So in this case also, they have used the talon and they have specifically targeted these genes. So this tells you that these genes are necessary for the B cell and T cell functionality. So if these genes are means affected, then it will lead to the immunodeficient zebrafish, which can lead to skid disease. So what information did we get from here? That this skid disease is linked to this gene. So this can tell us that which disease is linked to which gene is responsible for the disease. So in this way, uh, this tells you that skid is somewhere related to these PRKDC genes in the zebrafish. And this is the representation of that how the talon along with the FOC1 nucleases, it is leading to double strand break at this gene, and ultimately the result is the disruption of the gene, and the zebrafish is suffering with skid. So this is the F0 founder, and this is AP wild type after the microinjection of the capped mRNA into the yolk at one cell stage. So what we are doing with the microinjection, we are putting into the talon nucleus into the zebrafish, and then after this we perform the genotyping of this particular zebrafish after we have injected it with the talents. And after genotyping has been performed, then later on the phenotype is checked, that whether the zebrafish is surviving or is it showing the symptoms of the disease. So in this case, it was observed that the zebrafish which were injected with this talon, they were showing the symptoms of SCID. Okay, so this is all about the zebrafish SCID model. Then there are some knock-in rats also. So not just knockout, but knock-in could also be produced. So in knock-in, the study by Ponce de Leon et al. In 2014, they have introduced the point mutations. So what they have done is they have changed this A to T, A at 476 position to T into the NR3C1, which is the glucocorticoid receptor. So this glucocorticoid receptor gene, it has specific sequence. So at this particular position, they have just made one change in the nucleotide. Instead of A, they have changed it to T, just one single mutation. And then they have used the HDR donor plasmid with silent mutations to avoid the recutting. And they generated the precise knock-in rat models and demonstrated the potential of talons for complex gene editing. So in this way, with the help of talon, what they have done firstly they have looked into this gene which is responsible for the receptor site. They have cleaved it, and then they have specifically chosen the another DNA strand in which they have added the same gene but with this mutation. And means I'm not explaining again and again here, but what they are doing is simply they are having the homology arm sequences and then they are adding the gene in between it, so that the site where I've performed the double strand break using my this talons, so in that site I can change my genetic component of this NRC gene. So in my gene I'm having this particular sequence of this gene, so I'm changing it with the sequence which I'm now entering, which has this mutation. So I'm just doing the knock-in. So knock-in, but in knock-in also I'm bringing the mutation. So I'm replacing it. That is what is done in knock-in rats. So this shows you that not just this, but we can perform other complex events also in the case of by using the talons, we can also bring about the mutations or other changes also can be done. Yeah, and this is what they have shown, the talons, three left, three right. And then these are the restriction enzyme sites where the cut can be produced. And this is the site where the mutation has been incorporated. So previously it was G and now they have changed it to A. And similarly, in this also they have shown different cleaved sequences and different changes which can be achieved. So in the wild type it was G, but now they have changed it to A. And these are the results of the experimental products, that how the means when we want to check that whether the talon or all these nucleases are doing proper cleaving or not, because after the cleaving we should get two bands for the two different fragments. So we compare it with the reference set, that whether we are getting two bands or not, so that we can check that whether the nucleases are performing their work or not. And then we also will have the idea that where these nucleus can cut. So in that way, we will also have the information that these base pairs should be present. If that particular length is not achieved, that means the nucleases are not cutting the specific sites. Either there is some off-target binding. So these are that experiments which are done. So we'll not go into the detail, but this tells you that you can also use the talons for advanced uh, activities such as the knock-ins. And then these talents are also explored for the HIV therapy. So HIV requires CD4 plus CCR5 CXCR4 for the entry, which we have also seen in the previous session also, that the HIV virus binds to these receptors and then it can enter into the host cell. If these receptors are faulty, then the HIV cannot enter into the host cell. So people with this CCR5 del32, del32 simply means deletion of 32 nucleotides. So if that mutation is present, then that particular people are resistant to HIV. And why they are resistant? Because if this gene has the deletion of these 32 nucleotides, then this gene will not properly synthesize the protein which is necessary. So if protein is not synthesized, that means the receptor will not be formed and HIV will not be able to bind to that receptor to enter into the cell. So ultimately, people will get resistant to HIV. So talons are designed to mimic this natural knockout in patients' T cells. So these receptors are expressed on the surface of T cells. So generally, the talons are used in the T cells of the patients. So strategy is to disable these CCR5 receptors to prevent the HIV infection. So similarly, in ZFNs also we have seen the same thing. Similarly, can be achieved with the talons. So similarly, its fork1 unit will dimerize. It will bring about double strand break. After the double strand break, if it goes to the NHEJ repair system, then there will be uh, loss of some in between genes, means that will lead to the disruption of the gene. Some nucleotides might get deleted or they can join back in the improper ways. So some mutations can happen. So in that way, now the gene which is now after repair is faulty. So it will not synthesize the right protein. So which can help us to target the HIV virus. So this can make the person resistant to that virus because virus can no longer enter into the cell. Yeah, and this is the representation. So this particular um, receptor is the CD4 receptor, and these are the green ones are the co-receptor which are CCR5. So these both of them they act together. Now what we are doing is by editing it, we are simply bringing the double strand break in this receptor because these receptors are, these are the proteins and they are synthesized from the genes. So we are doing gene editing. We are cutting this particular sequence and we are cutting at the level of nucleotide. So if gene is cut, it cannot form the proper protein. So in that way, the HIV cannot fuse to the site and it cannot enter into the host cell. But if it is properly present, if there is no editing, then it can fuse and it can enter into the host cell. And then it can enter into the nucleus where it can integrate with the host DNA and it will synthesize the proteins which are essential for the replication of the viral DNA. And then ultimately, the virus will capture the host machinery and in the end, the host will die and the virus will survive. Okay. Then there is another study, which is the mRNA transfection of novel TEL effector nucleases, which facilitates the efficient knockout of HIV co-receptor CCR5. So this is the another way by mRNA transfection, and this is also known as CCR5 eucotelin. 2015 is the date when this paper was published. So this is the codon optimized talon. So UCO stands for codon optimized talons, and these targeted first intracellular loop of CCR5 receptor. So now there are different ways how you target. Either you disrupt the gene. In the previous session also we discussed that there could be multiple ways in which you can avoid the HIV to enter into the host cell. Either you can target the HIV genome or you can target the receptors where the HIV genome interacts. So similarly, in this case, they have generated the codon optimized talon, which can target the first intracellular loop of the CCR5 receptor. So it is delivered into the T cells by electroporation. So we have also looked into different methods in which you can incorporate these nucleases. So electroporation is one method, microinjection is the another, and then there are some other methods. Transfection is also one of the methods. So electroporation is one such method in which we are delivering the talon into the T cells, which results in the HIV resistant T cells generated in vitro. And this is the proof of concept for the talon based HIV therapy. So this tells us that how we can design the therapy using the talons, and it is simply targeting our um, first intracellular loop. So this one is the ICL1, which is the first intracellular loop. So it is influencing this loop and which is ultimately interfering with the HIV binding to the cell. Okay, so this is one of the way. Okay, now we will look into that other than talons, how the tail can perform different other functions. So the tail can also act as regulators now by using VP64 or CRAB. So if we use VP64, then it can activate transcription strongly. So for example, if I want certain gene to make multiple proteins, for example, if somebody is suffering from a disease in which some protein is not synthesized. For example, a person is suffering from a disease in which protein kinase is not synthesized properly. So what we can do is we can take the talons and we can target that gene so that it get activated and it can synthesize more and more proteins. So VP64 when bound to tail, it will activate the transcription and it will make sure that the gene is making more and more RNA. Sorry, in this case, it will be mRNA, and then ultimately it will give rise to protein. Then comes tail crab. So if crab is present, then it will repress the target genes. For example, if some gene is making a lot of proteins, such as oncogenes, so I don't want my oncogene to get activated. I want to repress it. So in that case, we can use the tail crab and we can repress that gene. And whereas in the VP64, if I have a tumor suppressor gene and person is suffering from cancer, then I can activate those tumor suppressor genes such as P53 is one example of the tumor suppressor gene. So this is how we can explore the tail for different functionalities, and it enables the precise control of gene expression without cutting DNA. Because for cutting the DNA, we need nuclease component. So in this case, we have replaced the nuclease with the CRAB or VP64. And this is the representation, then how it will look. So in this case, they have joined, instead of nuclease, they have joined VP64. So if VP64 is joined, then it will bind to my specific target site, the promoter region, and then this VP64 will bind to the transcription complex, which will activate the transcription of the target gene. And now how it is specifically binding to the target gene promoters, because we have designed the tail in such a way that it knows that where to bind in the genome. And then for repression, it is bound to the CRAB domain. And if it is bound to the CRAB domain, then there are some other mechanisms which will take place, which will not allow the transcription complex to synthesize here, and transcription will not take place. So if no mRNA is synthesized, that means no protein can be synthesized. So gene is repressed. Yeah, and this is also one such study. If you're interested, you can go through this. This also tells you that how the CRAB-based transcriptional repressions can be achieved, and this can lead to the knockdown of multiple gene targets. So I'll not go into the detail. This is just a paper. If you want to explore, you can go through this. Okay, now another purpose is the epigenome editors. So tail can also act as epigenome editor. So if tail is bound to DNMT3A, so then this particular component, it adds the methyl group. So if methyl group is added to the cytosines present in the nucleotide sequence, then it will silence the gene. It will not allow the genes to express. And then if there is TET1, then it will remove the methyl groups. Then it will reactivate the gene. So what I can do with this particular construct, that I can decide that when to express the gene and when to silence the gene. So we can study its expression. Yeah, so these are the different components which we can attach. Then comes tail histone modifiers such as acetylase, demethylases. So if I attach acetylase to my tail, then it can acetylate the histones. If I add methylases to my tail, then it will demethylate the histones. So in that way, we can reprogram the gene expression without altering the sequence. So we are not interfering with the sequence. We are not bringing mutations. We are simply performing the reprogramming. We are either doing the other, what you call them as, such as post-transcriptional changes. So in this case, the changes are performed at the level of nucleotides before the transcription. And then with that particular changes, we are determining that whether to express the gene or whether to silence it. And this also shows you the same. So this represents the DNA methylation. So DNMT, if it is bound to these particular tail elements, it is performing lot of methylation, and which is suppressing my gene, which is silencing my gene. But as soon as we use these TET1, then these TET1 will remove the methyl groups which are attached, and it will again reactivate this particular gene. And in this case, this is the example of epigenome editing. So these are the tail target site, and this is the area of interest. So these are the histone elements, and we can use histone modifiers. So this gives the example of TET1, which will remove the methyl groups. And then these methyl groups, in the TET1 case, the methyl groups are removed from the nucleotide sequence or from the DNA. Whereas if I use the histone modifiers, then they will remove the methyl groups from the histones. They can either remove methyl groups or they can add the acetyl groups or remove acetyl groups. So the example is LSD1, which is used to make the targeted histone modifications. So keep this thing in mind that methylation can happen at the nucleotide sequence level also and at histone also. So they are different methylations. So different proteins are used for that particular purpose. and both of them have different functionalities. So methylation of the nucleotides can lead to the silence of the genes. Then are mitotelins. So initially when we started with the session, then we discussed that although the talins are the predecessors of the CRISPR-Cas9 technology, but in the case of mitochondria, the talins are the best ones. Even the CRISPRs is not that better than talin. So mitochondria cannot be edited by the CRISPR easily because it requires RNA plus protein import. So the solution is to design the mitotalins, which means in the talins, they are fused with the mitochondrial targeting signal. So this mitochondrial targeting signal is what is helping these talins to enter into the mitochondria, and they are delivered as proteins directly into the mitochondria. So when we will talk about CRISPR system, then we will see that it also requires RNA, and that is the main challenge due to which we cannot use it in the mitochondria. So as in the case of talents, they can directly incorporate it in the form of proteins, and they can selectively cut the mutant mitochondrial DNA. So in this case, the patient's affected mitochondria, and these red circles, they are showing the mutants. So if lot of mutant sites are present, that means the person is affected. So this red circles means mutation heavy, means that that particular mutation is harmful for the mitochondria. So we can use the mitotelins or MTZFNs. So ZFNs is zinc finger nucleases. So that is the specific version of zinc finger nucleases which is targeting the mitochondrial mutated genome. So we can make the cuts in these mutated genome, and then partial elimination of mutated mitochondrial DNA, and then repopulation with the remaining mitochondrial DNA. So now in the end, the population will be with the improved mitochondria. It will have the right or the wild type mitochondrial genome. So most of them are removed. Means if we compare them, then compared to this patient with affected mitochondria, if we compare it with the improved mitochondria, then the number of mutated these genetic elements, they are less. So, and you know that why it is circular, because mitochondria has a circular genome, because our mitochondrial genome resembles the prokaryotic genome and it's not linear. That's why these circles represent the genetic elements. So this is how, by using the mitotalins, we can remove the mutated genetic elements from the mitochondria. Okay, and this shows that how the tail domains are binding to the this mitochondrial genome which is mutated, and then it is checking that where the mutation has taken place. So it can lead to the, because after the double strand break takes place, then we can add the desired um, DNA fragment so that it can undergo the homology dependent repair. So after that repair, we will add a fragment in such a way that it can bring about the mutation in the mutated segments. So in that way, we are repairing the mutated DNA element and we can convert it into the wild type element. That is one way. But other way could be that we can simply knocking it out. So that is also the other way, that we are simply removing them out. Okay, then there are some applications of mitotelins. So many mitochondrial disease, they are caused by the high mutant mitochondrial DNA load. So sometimes if the amount of the mutated DNA increases then as compared to the normal DNA, then the disease takes place. But in this case, as you can see, the mutated elements are comparatively less and the right ones are the high. So these little mutations can be still dealt with, but if the balance is disturbed, then that can lead to the disease. So that is what is shown here, that if the wild type mitochondrial DNA is greater than mutated, then the cells are fine. But as soon as the, sorry, okay, so this shows you, okay, so in the weighing weight, if the thing which is down, that means that is heavy. So in this case, the mutated mitochondrial DNA is heavy. That means it is, there are more number of mutated mitochondrial DNA as compared to wild type. So by using mitotelins, what we have done is that we have reduced the load of mutated mitochondrial DNA, and wild type DNA is now heavy. That means more of the DNA is wild type now, which is not mutated. So that is the advantage of mitotelins, that we can get back that balance. And mitotelins reduce the mutant DNA percentage below the disease threshold, that is less than 20%. So for example, if my percentage of the mutant DNA is greater than 20%, then that tells you that the disease is present, disease state. Healthy mitochondrial DNA will repopulate the cells, and how they will repopulate, because replication will take place. So the copy of mitochondrial DNA which is healthy will increase in the end. And therapeutic potential for the disorders like MELAS, LHON. So these are some of the disorders which are associated with the mitochondria. So these can be cured by using the talents. And this also tells you the same, the mutated mitochondrial DNA and the wild type. And this shows how using the mito-RE or mito-TELN or mito-ZFN, we can decrease the number of mutated mitochondrial DNA. And then in the end, it can recover to its original state, and number of the wild type mitochondrial DNA will increase, or you can say the normal or healthy mitochondrial DNA will increase. So these are the application of mitotelins. Then these tail can also act as the base editors. Now, if they are fused with the DDDA, cytidine deaminase toxin A, this performs direct C to T or G to A edits on the double-stranded DNA. So that is what we are doing here. This can also act as base editors. That means we can change the nucleotide bases. So either from C to T or from G to A on the double-stranded DNA, and it can work inside the mitochondria also. So tail base editors can also be used inside the mitochondria, which we have seen previously, that not just disrupting that particular genetic element, but we can also correct that mutated DNA, which is present inside the mitochondria, by bringing about these mutations by using the tail base editors. And this split DDA halves reduce the toxicity. UGI fusion increases its efficiency. Yeah, and this is what is shown here, that how the conversions can take place. So tails can also act as the base editors, and this is the DDDA cytidine deaminase toxin A unit, which is attached to the these tail effector molecule. Okay, and the composition of tail base editor. So this is the composition. It has UGI, it has DDA, NC site, and recommended DDA halves. So this is the toxic element in this DDA. If we explore it, if we increase, if we zoom it, then how it appears. So it has some toxic component to it, and then this DDA arm which is present. So it depends that what kind of mutation will take place depending on the this DDA tox halves, which will determine that what kind of mutation will take place. So it can either be C to T or G to A, depending on this particular DDA tox halves. Then comes tail transposases. So these domains can be fused with the piggyback transposase domain, and in theory it allows the insertion of DNA cargos at chosen sites. So generally transposition means jumping genes which can move from one place to other place. So in this also, by fusing the tail with the piggyback transposase, we can add some DNA cargo at particular site. We can add particular gene at particular site, and observed insertions usually within the 250 base pairs of the target. And current limitation is that it has very low efficiency. So this is one of the limitation of transposase, but this tells you that they have the potential to add something, some genetic element. So not just entire gene, but some smaller DNA fragments, that is known as, in this case, they are calling it as DNA cargo. So as you can see, these are just four nucleotides. So four nucleotides are added before and after the transgene with the help of these piggyback transposase. And this is also the another representation, that from this particular construct, with the help of this piggyback, what I'm doing is I'm adding some component in between, which is from the CMV. So coliform mosaic virus or some particular element, genetic element I'm picking up from the another region, and I'm putting it inside my target gene. So it could either be inside the target gene or near the target gene. So this is what is the work of the transposases, that I can insert any genetic element at the target site or near the target site or desired site where I want to add it. So this is the purpose of tail transposases. Then tail recombinases also are present. So in this case, tail domains are fused to the recombinase, for example, GIN, FLP, and these allow the targeted genomic rearrangement such as inversion or excision, and require conserved recombination motifs in the DNA, and this have potential for synthetic biology and genome editing. So as we know that in the case of FLP, they have FRT sites, and depending on the orientation of those FRT sites, whether they are facing towards one another. So facing means that they have a sequence. So what is the directionality of the sequence? So if I'm reading it from 5' to 3' direction, then the other sequence is also read in the same direction, or is it opposite? So if they are read in the same direction, then it can lead to the deletion. If they are in the opposite direction, that means they are facing each other, then there will be inversion. Then these are the FRT sites which are recognized by FLP recombinase. So based on the orientation of these sites, it depends that what will be the output, whether it will be the insertion or deletion, sorry, inversion or deletion. And this is also one of the example, FLP TAL. The TAL module is responsible for delivering and stabilizing the FLP module onto the desired genomic FRT like sequence where the FLP module mediates the recombination. So this thing also need to be kept in mind that wherever the FRT sites are present, we need to see the nearby sequences which are present so that we can design TAL effector in such a way that it can bind to those sites and it can stabilize my FLP variant such that FLP variant is binding to the FRT sites, and then it will bring about the function. So depending on the orientation, that whether it is facing each other, it is facing on the opposite direction, so output can vary, which we have discussed in this case. Okay, now this is the summary of what we have talked about talents. So talents are the transcription activator like effector nucleases, and these are used by the scientists to improve the food crops. So this is the in terms of agriculture, not just food crops, but there are other applications also. But this is were the initial findings when it started with the rice, that they worked on the rice to increase the yield. So that is how the talents was started. They have started to explore talents, and these are natural proteins. So tails are the natural proteins uh, which are used by the harmful plant bacteria such as Xanthomonas, and so that they can infect the plant cells. So tails, we are only talking about tails and not the nucleases one. So tails are those proteins, and then scientists have used the discovery of tails to modify the tails into talons. Now what they did is they added the nuclease component also into it, and they have also designed these talons to target the selected or specific DNA sequences, which we have seen that they can bring about the knockout at particular sites or knock in at particular DNA region. So that is one thing which the scientists have done. They have made cut at the targeted DNA sequence. They have initiated the cell's on repair mechanisms to remove the new DNA at the target site. Now, how these talons are different from CRISPRs? So, talons are extremely precise gene editing tools which can target any sequence and perform better than CRISPR in some cases. For example, in the case of mitochondria, where we discussed that it is difficult to deliver the CRISPR RNA into the mitochondria, but in case of talons, we can simply put them in the form of proteins into the mitochondria. So they are better as compared to the CRISPR in terms of mitochondrial uh, genome. And these have additional capabilities to perform nucleic acid free editing. That means they do not use any RNA sequence or any other sequence. They in themselves are sufficient enough. For example, in CRISPR Cas9, the Cas9 is dependent on CRISPR and guide sequence. But in this case, they are complete in themselves, so they do not need any nucleic acid sequence for editing. And they can discriminate between the DNA modifications like methylation, which affects the gene expression, and they can modify the DNA within the organelles, which we have already discussed, such as mitochondria and chloroplast. Now, how they are used to improve the crops. So, what they can do, they can improve the traits. And as we know that there are different genes which synthesize different proteins, and those proteins will determine the output of the plant, that whether it will give high yield, low yield, what will be its taste, how it will fight against disease, pathogens, climate. So all these things are governed by proteins. And what we can do with the talents, we can find those genes which are responsible for these traits, and we can specifically target them. We can either perform the knockout of those genes or knock in of those genes, or we can change their expressions. So that is how we can use the talents to improve the yield, we can improve the taste, we can make them resistant to disease and pests, and we can also design such crops which are resistant to the drought conditions or salinity conditions, and we can also improve the quality. Then examples of talents are soybeans that produce premium quality oil, and then rice, which is more aromatic and resistant to the bacterial blight. So these are the products which are achieved after the talents. And then potatoes with better taste and less browning and lower levels of potentially carcinogenic acrylamides. Then wheat with the complete resistant to powdery mildew. So these are some of the examples that where the talent was used to improve the crops. And how to gain access to talent technology. So there are certain sites from where the talent you can get access to talent technology. So one such as Two Blades, which give no-cost talent rights to non-profit and multilateral entities such as International Ice Research Institute to facilitate the improvement of crop varieties and support the innovation to benefit the food security. Yeah, so this is all about the talents. Now we will start with the question answers, but if you have any doubts, then you can ask. Okay, now I'll start with the question answers. So first question is, which system is responsible for secreting the tail proteins into the plant cells? Option A, T1SS, option B, T2SS, option C, T3SS, and option D is T6SS. Okay, so yes, you both of you have given the right option. So which is T3SS. So we have seen that how with the help of T3SS, the xanthomonas is allowing the TAL effector to enter into the plant cell, and this is the structure that how it appears and what are the constituents of this particular this protein, this particular element which is responsible for the secretion. Second question is, which gene is primarily targeted by the PTHXO1 tail protein in rice? Option A, OS sweet 11, option B, OS O9G 29100, and option C is OS sweet 14, and option D is OS sweet 13. So this might be confusing because we have only discussed that sweet gene is targeted, so it's all right. Okay, so I mentioned option A. So yes, option A is right. So that is OS sweet. So OS stands for um, okay, so sorry. So I'm Oriza sativa. I'm not properly able to pronounce it. So that is the species name of the Oriza sativa. So for the rice, so that OS specifies that, and sweet means the gene which we are targeting. And then similarly 11 and 14 and 13. So these are some of the numbers which have been given. So in this paper, which is obtained from the molecular plant, I have shown that these tail proteins, in this case it is PTHXO1. So the thing which I have highlighted in green, this box, so this shows you the PTHXO1, so it targets the OS sweet 11p gene, whereas if we talk about PTHXO2 tail protein, it will target OS sweet 13p gene, and similarly this PTHXO3, it targets the OS sweet 14p gene. So for this particular tail protein, OS sweet 11 is the right answer. So yes. Okay, next question is, what is the primary function of talents in genome editing? So option A is protein degradation, option B is gene expression inhibition, option C is DNA cleavage at specific sites, and option D is RNA interference. Yeah, so you're right, because we know that nucleases have the ability to cleave the sites. So that's it. This is the right option. Okay, so next question is, which enzyme is most commonly fused with the tails to create talons? This is also straightforward. Option A is EcoR1, option B is FOC1, option C is VAMH1, and option D is HIND3. Yeah, so FOC1 is the enzyme which is most commonly used as compared to the other ones. And this is the structure of FOC1. Yeah, and this particular sequence shows you the RBD domain, RBD region, which is present inside the central repeat domain. And there are total 18 such repeats. Okay, next question is, which model organism was used for the generating SCID models by a talent-mediated knockout of PRKDC gene? Mice, option B zebrafish, option C is rats, and option D is goats. Yeah, so this is also straightforward. We have discussed that zebrafish was used as the model organism for generating the knockout of the PRKDC gene. In this particular construct, what they did was that they have used talent, which specifically disturbed this particular PRKDC gene, and this gene was responsible for the B cells. And if B cells are not synthesized properly, then ultimately the immune system will get compromised, and the zebrafish will generate the skid symptoms. Okay, next question is, which talent assembly method uses the hairpin capping oligonucleotides to improve the assembly efficiency? Option A, flash assembly. Option B, iterative capped assembly. Option C is Gibson assembly. And option D is star protocol. Yeah, yeah, so this is the right option, iterative capped assembly, because we have discussed that in the case of iterative capped assembly, there is a oligonucleotide cap, or sometimes it is also known as the hairpin, with which it avoids the incomplete products to bind to the newer units. So we have also discussed the reason that why it is important, and this is well suited for constructing the error-free tail arrays, because if capping does not take place, then in the scenario, for example, in this scenario, if it does not take place here, then the B which is coming, it will bind here and A is missing. Similarly, here also, if the capping does not take place after the A, then the C will bind to A and it will assemble the faulty nucleotide, faulty talon repeat units. So to avoid that, capping is important. Now let's move to next question. So what is a common outcome of talons targeting mitochondrial DNA? Option A, HDR based recombination. Option B, NHEJ mediated repair. Option C, rapid degradation of mutated mitochondrial DNA. And option D is activation of nuclear encoded mitochondrial genes. So yeah, you have mentioned right. So option C, rapid degradation of mutated mitochondrial DNA. This we have already seen that how in the patient which were affected, they have the higher number of, or if we if we compare the balance, then the numbers of mutated DNA was high as compared to the normal DNAs. And with the help of mitotalins, we have disrupted those units. So now we have less of mutated DNAs and more of the normal DNAs. And now with time, these normal DNAs will replicate, they will increase their numbers. So the person or the patient will have the improved mitochondrial DNA after some period of time. Okay, and if we look at the other options, such as HDR based recombination, so the mitochondria lacks this efficient HDR pathways. And if we talk about NHEJ mediated repair, so mitochondria do not support the classical NHEJ repair. And if we talk about option D, which is the activation of nuclear encoded mitochondrial genes, so this is also means incorrect, because talon is only acting on the mitochondrial DNA and not on the nuclear gene activation. Okay, next question is, which gene was targeted in talon-based knock-in experiments in rats for studying the glucocorticoid receptor functionality? Option A, NR3C1, option B, IgM, option C is PRKDC, and option D is CXCR4. Okay, so it's related to glucocorticoid receptors and not related to the SCID. So let's see. So the right answer is option A, which is NR3C1 and not PRKDC, because we have seen this gene in the zebrafish construct where they were studying SCID models. And this particular NR3C1, this is targeted in the talon-based knock-in rats when they have to study the glucocorticoid receptor function. And this is the paper in which they have in detail mentioned that how they have achieved it. Whereas if we talk about IgM, then this was targeted in the knockout in the case of rats, where they have seen that if we target this gene, then the B cells and antibodies were not synthesized. Okay, and similarly, the CXCR4, so we have seen it in the case of HIV. Okay, next question is, which mechanism is used to achieve high targeting specificity with tail-based editors? Option A, splitting the deaminase into the two non-toxic halves. Option B is using full-length DDDA domains. Option C is co-delivery of talon and RNA components, and option D is incorporating a fluorescent reporter. Okay, so you have mentioned option B, right? So the right answer is option A, which means splitting the deaminase into the two non-toxic halves. So the question was that how to achieve the high targeting specificity with tail-based editors. So when we were discussing about tail-based editors, then we discussed that the DDDA domains which are used, they split into the non-toxic halves. So that particular splitting will decide that what they means what mutation they will bring. So the splitting the deaminase into two toxic halves. So these use the split DDA halves that only becomes active when brought together at the target site, which ensures the high specificity. And next option was using full length DDA domains. So this is incorrect because the full length DDA domain is highly toxic to cells. And then next option was co-delivery of TELN with RNA components. So this is also incorrect because these editors are protein-only systems. It is not like CRISPR, which uses the guide RNA. So this is not the right answer. And then incorporating a fluorescent reporter. So this is also incorrect because reporters can help you track the editing, but that will not increase the specificity of the binding. So next question is, what is the main advantage of using TELNs over ZFNs for genome editing? Option A, smaller cDNA size. Option B, lower cost and ease of delivery. Option C is a simpler protein DNA recognition code. And option D is higher specificity and lower off-target effects. Okay, so the right answer is option C, which is the simpler protein DNA recognition code. So let's look at all these options. So first option was that smaller cDNA size. So this is incorrect because it has larger cDNA compared to zinc finger nucleases. And then lower cost and ease of delivery. So this is also incorrect because talons are large. Because we have when we will compare the structure of ZFNs and the talons, then in talons we have already seen that 18 such repeat sequences are again and again repeating, and each repeat is 34 amino acid long. So talons are larger. And in zinc finger, we only had the triplet of the amino acids which were binding to one single nucleotide. So ZFNs are short as compared to talons. So that means they do not have low cost. They will definitely have high cost as compared to ZFN. Then third option was simpler protein DNA recognition code. So this is correct because talons follow a straightforward one repeat, one base RVD code, unlike the complex triplet recognition of ZFNs. So in this case, it was straightforward that the two amino acids will bind to one base, which are present inside each repeat. So this is the right answer. And then D option was higher specificity and lower off-target effects. So this is also partially true, but the primary advantage is this simpler protein DNA recognition code. That's why option C is correct. But we cannot say that option D is completely wrong. Option D is also right, but C is better option than option D. Okay, so next question is, which of the following best describes the N-terminal region of tail proteins? Option A, it contains the nuclear localization signals and a secretion signal which is required for the delivery into the host cells. Option B, it forms the DNA binding repeats that directly determine the base recognition. Option C, it carries the acidic activation domain which is required for the transcriptional activation. And option D is it acts as a cleavage domain for the non-specific DNA degradation. Okay, so you mentioned option B. So let's see. So the right answer is option A. So this N-terminal region. Okay, so yeah. So it contains the nuclear localization signal and the secretion signal, which is required for the delivery into the host cells, which is present towards the N terminal. And simply, if we look at option B, then it says that it forms the DNA binding repeats. But DNA binding repeats is not at the N terminal, that is at the central repeat domain, which is the CRD. Each central, there were total 18 such repeats, and in each repeat, there is RVD, that is the variable sequence which was present. So variable sequence of two amino acids, which is responsible for binding. So that is not the N-terminal part. That's why option B is not right. And option B is right, sorry, option A is right because it carries the nuclear localization signal. And we have seen that from xanthomonas, when these tail elements enter into the host cell, it enters into the cytoplasm, so it is non-functional at cytoplasm, so it needs something to enter into the nucleus. So that particular signal is present at the N terminal. If we talk about acidic activation domain, then this acidic activation domain was present towards the C terminal end, which was responsible for the transcriptional activation. And whereas if we talk about it acts as a cleavage domain, so this is also not right, because for the cleavage we have the nucleases and not the tail. So next question is, which of the following statements correctly describe talons? Option A, talons consist of tail DNA binding domains fused to catalytic domain of Cas9. Option B, talons generate DNA. Okay, so yeah, you are right, but the N-terminal region of tail protein, yeah, that is also, so what happens is that whenever a protein is present in the three dimension, so in that case, it doesn't mean the N terminal will not be interacting with the C terminal region. So in this case, the N terminal region, which is present towards the N terminal of the protein, so when it will adopt the structure, so in that time, the N terminal region will be interacting with the that AA acid activation domain also. But the main thing which is determining its nuclear localization, that is present towards the N terminal region. And the secretion signal is also lying at the N terminal region. Whereas this acidic activation domain, this is somehow helping it, and along with this acidic NLS signal was also available when we have seen the diagram. So maybe after completing this, we'll look into that figure again. So although the signals were present there, but this N terminal is also governing the signals through which the tail protein get into the nucleus. Okay, so next question is, which of the following statements correctly describes the talons? So option A is talons consist of tail DNA binding domains fused to the catalytic domain of Cas9. Option B, talons generate DNA double-strand breaks through dimerization of fork-1 nucleus domains. Option C is talons function by directly recognizing RNA secondary structures instead of DNA. And option D, talons act as the transcriptional activators without altering the host genomes. So you have mentioned option B, right? Yes, option B is right. And we have already discussed it a lot of times that the dimerization further on leads to double strand breaks, whereas other options are not right. Next question is, how do tail proteins recognize the DNA target sequence? So option A, by forming the hydrogen bonds between their beta sheets and the major groove of DNA. Option B, by aligning their tandem repeats where each repeat specifies one nucleotide by its RVD. Option C, by binding to RNA intermediates that guide them to DNA. And option D is by using the random charge interactions across the phosphate backbone. Okay, so you mentioned option B. So yes, option B is right, because we have already seen that how this particular, for each repeat there is RBD sequence which will bind to one specific nucleotide sequence. And if we look into the option A, then they are saying hydrogen bonds. So hydrogen bonds takes place, but one of the residue, which is the 13th residue, which is responsible to interact with one of the nitrogenous base, and this option is completely wrong. Using this, they are saying beta sheets and the major groove of DNA, so this is not right. And then in the C option also, they are saying that by binding to the RNA intermediates. So there is nothing like RNA intermediates. The talons are completely nucleotide free units, so they do not require RNA intermediates as like CRISPR. And then the last option was that by using the random charge interactions. So this is also incorrect because there is nothing to do with the charge. Next question is, which of the following best explains why talons must be used in pairs? Option A, single talon cannot recognize the specific sequences without the partner. Option B is fork-1 nuclease required dimerization to cleave both strands of DNA effectively. Option C, DNA binding domain of one talon stabilizes the transcriptional activation domain of the other. Option D is talon dimers are required to recruit host transcription factors for the DNA recognition. sorry. Yeah, so this is quite straightforward. And as you can see, many of the questions are kind of repeating the same information. Yeah, so this is the right answer, and whereas other options are not true. Now, next question is, what is the function of nuclear localization signal in the tail proteins? Option A, they allow the tail proteins to self-replicate in the host cytoplasm. Option B, they target the tail proteins into the plant nucleus where they act on the host DNA. Option C, they recruit the acidic activation domain to the DNA bound regions. And option D, they stabilize the tandem repeat array during the DNA recognition. Okay, any guesses for this question? Have you mentioned option B for this? Okay, so the right answer is option B, that they target the tail proteins. As the name suggests, nuclear localization signal. So it is clear from here that this particular signal is somewhere essential for the proteins to enter into the nucleus of the plant cell, whereas other options are not true. Yeah, other options are not hinting towards the same direction. Okay, so next question is, which region of the tail proteins directly determines DNA base specificity? So this question is also again repeating. So option A is the acidic activation domain at the C terminus. Option B, the repeat variable diresidue RVD within the each repeat unit. Option C, the N-terminal secretion signal peptide. And option D is the nuclear localization signal. Yeah, so it's straightforward. Option B is the right, which is the RVD particular diresidue sequence present inside each of the domain of the central repeat domain region, which is also referred to as DBD, that means DNA binding domain. Okay, so next question is, what structural features? Yeah, I didn't realize earlier, but yeah, now I'm also thinking the same. Each and other option is B. So next question is, what structural features give tail proteins their modular DNA recognition ability? Option A, a beta sheet domain with the random residue variation. Option B, tandem array of 33 to 35 amino acid repeats, each recognizing one nucleotide. Option C, a zinc finger domain that interacts with trinucleotides. And option D is an RNA binding loop that indirectly guides the DNA binding. So here they are talking about the DNA recognition ability. So again, okay, so in this case also the answer is option B. Yeah. Okay, so Tikeshwari has mentioned option A also. Okay, so one of the another thing which I want to mention is that when we were looking at tail proteins, then we discussed that in tail proteins there is the two alpha helices, helix bundles, and then in the middle there is a loop, and that loop is for the residue 12, 13, which are responsible for recognizing the nucleotide. So there is no beta sheet domain like structure present. So that's why option A could not be right. And option B is right because we have seen that in each repeat there are 33 to 34 amino acids which were present. And in that particular repeat, there is RVD which recognizes a single nucleotide. Then option C was related to zinc fingers, and option D was related to CRISPR technology in which RNA binding loop is present. Okay, so next question is, in TALEN technology, why is fork1 nuclease fused to tail domains instead of being used alone? So option A, fork1 alone cannot enter the nucleus of plant or animal cells. Option B, fork1 requires tail repeats to provide sequence-specific DNA recognition. Option C, fork1 is naturally inactive unless it binds to RNA. And option D is fork1 function only when stabilized by acidic activation domains. Yeah, again, option B is right. So I don't know, I didn't check it earlier, but yeah, all the B options are only right. Yeah, so this is right because the option A says that fork1 alone cannot enter the nucleus. So fork1 has nothing to do with entry into the nucleus of any plant or animal cell. So this is completely wrong. Option B is right because we know that tail is the DNA binding region or DNA binding domain. And we only need this tail because we want to specifically target some genes. Similar thing with the ZFN also. In ZFN also, the zinc finger domains which were present, they were also to specifically bind to the nucleotide sequences of our interest. So in this case, tail is also having the same function, and fork1 can have can perform the nuclease activity, but it can perform nuclease activity anywhere. So to target it to the specific site, we use the tail repeats. And then they are saying that fork1 is naturally inactive unless it binds to RNS. This is also completely wrong because these fork1 nucleases, they bind to the DNA. And the last option was that fork1 function only when stabilized by acidic activation domains. So this is also wrong because acidic activation domains are not responsible for the fork1 function. Okay, so now let's move to the next question. So which of the following is the key difference between the tail proteins and the zinc finger proteins in the DNA recognition? Option A, zinc fingers recognize the single bases, while the tail repeat recognize the trinucleotides. Option B, tail repeats recognize the single DNA bases, while zinc finger recognizes the three base segments. Option C, zinc finger recognize the dimerization, while the tails do not. And option D is the tail proteins cannot be engineered, while the zinc fingers can. Okay, so you have mentioned option A. Okay, so again, option B is right because we have seen that zinc finger recognizes the. Okay, so they are saying that zinc finger recognizes a single base, while tail repeats recognize the trinucleotide. But we have seen that tail repeats recognizes only the single base and not the trinucleotide. So this is not right, and that's why option B is right. And similarly, zinc finger also recognizes the three base segments. So as you can see, each of the zinc finger domain is binding to three nucleotides. And then option C was that zinc finger require dimerization while tails do not. So this is also wrong because both of them require dimerization of their nucleus domain to function. And then tail proteins cannot be engineered while zinc fingers can. So this is also completely wrong because we can engineer tail proteins also, which we have seen different steps that how we can perform the modular assembly of the tail proteins and zinc finger proteins. So option B is right. Yeah, and this is the representation how both of them appears and how they are different from one another. Now the last question is what role, maybe last question. So what role does the acidic activation domain at the C terminus of tail protein play? Option A, it initiates DNA cleavage during infection. Option B, it recruits the host transcription machinery to activate the gene expression. Option C, it helps stabilize the tandem repeat structure. And option D is it provides the nuclear localization for DNA entry. Okay, yeah, so the right answer is again option B. Okay, now I guess um, you will be completely confused related to the nuclear entry. So yeah, now we will clear this concept. Yeah, it's again B. Uh, because I have picked these MCQs from different sites, and in all of them, maybe B option is right, and I've arranged them in such a way. So, okay, now let's look at each option one by one. So, they are asking for the acidic activation domain. So, now here it's the structure of tail monomers. Now, we will clear the doubt of N terminal and nuclear localization signal. So, you were right that the nuclear localization signal is present towards the C terminal of the protein, and then this is the targeting sequence, which is the TS. Now, this NLS and TS. So, to deliver the tail into the nucleus, there is the need of both the targeting sequence and the NLS sequence to take place. So, they both will interact, and this will target the NLS. This will target the entire tail into the nucleus. But NLS signifies the nuclear localization signal. It is having the signal which is telling you that this particular protein is the meant to be enter into the nucleus. That is what is the role of nuclear localization signal. But what thing will read it and then means move it into the nucleus? For example, in our body we have Golgi apparatus, which is having the role of a postman, so which decides that where the protein should go, whether it should go into the cytoplasm, it should go into the nucleus or some organelle. So similarly, in this case also, the targeting sequence, which is TS, that will decide from NLS that whether to means enter this tail into the nucleus or not. So that's why in that case, N-terminal region confusion arised, because this particular sequence is helping the this delivery of the tail into the nucleus, but the signal for the nuclear localization is present towards the C-terminal region. So that is right. Okay, so first option was that it initiates the DNA cleavage during infection. So that is not true, because there is no cleavage which take place if we talk about tail. And second option was right, that it recruits the host transcription machinery to activate the gene expression, which is the main work of this domain, because it will activate the genes such that it can help the bacteria to infect. It will provide the nutrition to the bacteria to survive. And option C was that it stabilizes the tandem repeat structure. So this is also not true. And then option D was that it provides the nuclear localization for DNA entry. So it do not have any role with the nuclear localization, and it is not the activation domain. So yeah, that's it. Okay, we have other question also. So next question is, in genome editing applications, what ensures that the talons create the targeted double strand breaks? Option A, their tandem repeat array bind the RNA molecules before cleavage. Option B, in each talon pair binds opposite strands, positioning fork1 domains to dimerize. Option C, the acidic activation domain guides fork1 to DNA cleavage sites. And option D is talons randomly bind DNA until the cleavage occurs. Yeah, again, option B is right. So, yes, because we already have discussed it a lot of times that fork1 will dimerize and then cleavage will take place. Okay, so this was all. But yeah, so I hope you have got the clarity about the talents. And if you still have any doubts, then you can ask. Or if you need any other material, then also you can let me know. Yeah, so this is all about today's session. And then in the next session, we will be discussing about the CRISPR-Cas9. No, CRISPR-Cas9 will be in the next class. That is week 7. In the previous class, we have discussed zinc finger nucleases. Okay, so that's it. So I'll end the class. Thank you very much for being here, and we'll meet in the next class. Thank you. I'm ending the class.